In today's class, we will study preclinical research methods in drug development. This topic is present in BMS third phase syllabus in that research subject and in the fifth chapter, eighth topic. Now here the topic is preclinical research. What is meant by preclinical research? It means that before giving to the human being, the new trial drug is tested on the animals or the other in silico in vitro. That is called as a preclinical methods. Here the majorly four methods we are using to test in the form of in silico method, in vitro methods, in situ as well as in vivo methods before giving to the clinical or human being. Now we'll study what is meant by preclinical. Preclinical research is the initial phase in the drug development conducted before trial. It means that the before giving to the human beings, this is one of the initial phase of the drug development, the new trial drug. Here, what are the things here we are evaluating? First is the safety of this, whether the drug is safe or not, efficacy, whether the drug is effective or not, pharmacokinetics, it means the ADME we are studying here, toxicity, whether drug is toxic, of new drug, the candidate that we are testing here in the methods of such as the in silico methods, in vitro, in situ, in vivo studies we are conducting in this phase. Now we'll study one by one what is meant by in silico method. The first term is in silico. In silico is nothing but this is the computer chip is called as a silico. In silico is nothing but the inside the computer. It means without using any humans or animals, that only by using the computer or only the using the computer chip, we are predicting the drug, how it is effective, how the drug is working, that we will study in this phase. In silico means the analyzing the computer based studies. It means that is analyzing the predicting how the drug will affect. The principle is use of computational stimulation to predict drug is target interaction, ADME properties and toxicity. Before other methods using this drug is firstly we are using for predicting the target interaction, the what is actual where you want to target, the whether the drug is targeting or not, that we are analyzing the interactions, the combinations as well as ADME, the absorption, distribution, metabolism and excretion property also we are checking through the computer, as well as the toxicity also we are analyzing using the in silico method. Okay, and now we can take very good example here, the molecular docking of curcumin. You know that is the curcumin is one of the extract that is curcuma longa. In simply it is called haldi or haridra in local language, haldi. In that the phytoconstant is present chemical, that is curcumin. Now how we are using this computational technique, that is in silico methods, you know that is whenever there is a body in that inflammation it is there, the COX-2 enzymes is the due to the COX-2 enzymes the inflammation you can see. So whether the if you give the this curcumin, whether it is effect on the COX enzyme or not, that we are predicting by giving the analyzing in the computer. Just we are entering the curcumin here. Docking is nothing but the combining. The molecule of curcumin is combining with the this in the computer based studies. Here the COX with COX enzymes, you know that is the COX-2 enzyme is a one of the inflammatory enzymes. Whenever the inflammation is there in the body, this enzyme is the responsible enzyme to predict the inflammatory activity, whether the curcumin is the helpful for the acting as an anti-inflammatory drug or not, that we are analyzing using this computer technique. This is the one of the very good example in Ayurveda. Okay, now the next one is application where we are using this in silico method. The mainly the molecular docking. Just now I told the molecule is nothing but the new trial drug. Here the docking is nothing but the whether the drug is the uh, combining or interacting with the our target that we are analyzing. It means that whether it is correctly working or that is a combining it not, that is we are checking, that is the docking. Next is the visual screening. What is meant by visual screening? The lots of the molecules we are passing through this in this computer, which drug is that is combined with the particular target, that drug we are using for the next stage or for the animal trial or human trial. It nothing but the visual screening is nothing but the so many thousands or lakhs of the drugs we are searching in that, which drug is in the computer shows the effective, that we are separating and using. This is the visual screening. Next one is the QSAR modeling. What is meant by QSAR modeling? It means that quantitative structure activity relationship. It means that the similar drugs we are using, very good example that omeprazole is one of the PPI drug. The similar structure the scientists use to produce the pantoprazole. Okay, like that, from the pantoprazole again rabeprazole, esomeprazole, like that the similar structures the scientists using for new drug development, for the development of new drugs. This QSAR modeling technique, we are using that similar structure drug, something only little bit will change whether drug is effective or not, we are analyzing by using in silico method. This is also one of the best application through the in silico method. Next is the advantages. Why the in silico method we are using in the research? The mainly it is a rapid, fast acting, ethical. There is no need for permission for any from anywhere, animal committee, ethical committee, or human ethical committee, and cost effective. That is does not require much money to for spending. This is a very cheap. Limitation is the based on the models, because this is only we are analyzing based on the models only. That's we are not at all using the any living organism. That's why this is one of the limitation. It is there may lack of full biological accuracy. It may be not accurate like the any other trial. That's why these are the limitation in the in silico method. This is about the in silico method. Okay, now we'll study the next one, the in vitro method. In vitro method is nothing but that is outside or in the glass or glass tube or test tube, or we can call as a petri dish, like that, something the cells we are separating. What are the things here we are using in vitro methods? Specifically cells, enzymes, or any tissues we are separating from the body, that is stored in the lab in the controlled environment. In that controlled environment we are giving our new trial drug. Whether the drug is acting or not, we are checking in the glass slide or petri dish. In that we are analyzing. This is technique is in vitro method. Here lab based study. This is the preclinical lab based study here performed outside the living organisms. It means that we are separating the here cells we are separating, or else tissues or enzymes we are separating and using in the controlled environment in the lab, specific environment. We are analyzing whether the drug is acting or not. Example we can take, that is the ashwagandha withenia somnifera root extract. That root extract we are separating here, tested on human cancerous cells to assess the cytotoxic activity. What it means? The simple meaning is that the in vitro methods, the cancerous cells we are separating in the test tube or petri dish, that we are keeping in the controlled environment. After that, the ashwagandha withenine is phytoconstant. We are separating this chemical constituent, we are separating and adding in the this cancerous cells. Whether the drug is effective or not, we are checking in the in vitro. If the drug is kills the cells, or the drug after the using of this quinine, the drug, the cells, enzymes or tissues, there dies, it nothing but the, this is one of the cytotoxic drugs. It means that you can use the drug in the cancerous cells. Like that, we are analyzing the in the lab, in the petri dish or in the test tubes, we are checking whether the, if you are using this drug, whether it is safe for the cells or tissue or not, we are checking here in this methods. Okay, this is the example. Next is the applications. Here application is the cell viability assays. What is cell viability assay? Just now I told whether the your new trial drug is safe for the cell or not, that we are analyzing by using in vitro method. This is the cell viability. Next one, enzyme inhibition study. You know, that is the whenever we are separating the enzymes, the most of the, due to the enzyme, that so many diseases are produced. Whenever the new your trial drugs, the if you are giving, here the checking the enzyme inhibition, the drug is inhibit the enzyme or not, that we are studying by using in this method, in vitro method. Next one is the receptor binding assays. This is a very important method for analyzing the in the research, the receptors binding. It means that, you know what happened, whenever you are giving the your trial drug, your main important the trial drugs, whether it is binding with the this receptor or not. If binding it, then only it will work. That also helpful for the assess or analyzing in the in vitro methods, that your drug is whether binding with the particular receptor or not, that we are analyzing in this method. This is the in vitro methods. Now advantages, what is the here advantages? High throughput it is there. It means that the so many things we can do at a time. This is a very high thoughtful process you can do with the in vitro method. It means the fast you can, at a time you can do so many high thoughtful process. And next, reduce animal use. No, that is the very problematic, that using animals or human beings. This is the one of the safest, the reduce the animal use here because of this in vitro methods. Now the limitation, what are the limitations? Why some people they are not believing here? It does not replicate full body interactions. It means that only you are separating the cells, uh, tissues or enzymes only. We cannot completely tell that whether drug is acting or not, because only simply the we are testing on the only simple cells. That's why it is does not replicate full body interaction. This is one of the limitation in the in vitro method. The next one, third one, is the in situ methods. What is mean by in situ? In situ is nothing but the particular location or inside the location. Here in situ means the tissue level study. Tissue level study is nothing but as it is in their physiological conditions we are keeping, in that only we are giving our trial drug. This is called as a in situ methods. Here in situ method is conducted on organs or tissue in their natural position to maintain the physiological structures. You know, that is as it is, how it is the naturally present in the human beings. Similarly, they are separating and they are keeping in their natural the positions and maintaining the physiological structure. After that only we are doing the research. This is the in situ methods. In situ is nothing but, situ is nothing but their location, inside the location, as it is the locations we are doing experiment here. The example is isolated rat intestine used to study the effect of the eagle marmulose extract on the intestinal motility. You know, that is bilwa, that is eagle marmulose, bale patra is the common drug we are using for the loose motion or diarrhea, dysentery. In the Ayurvedic, commonly they are using Ayurvedic physicians in the loose motions to show their property, whether the drug is acting or not. That if you want to prove, you can use this in situ methods. What here the experiment is that isolated rat intestine. First of all, that rat intestine we have to separate. That intestines as it is to physiological structure, the natural position you study in that intestine. We have to add the bilva, eagle marmulus extract on intestine, whether the its intestinal motility, is there any changes or not, that we are analyzing. This is the one of the very good example of the this in situ method here in the preclinical methods. Next is the applications. Application where this in situ method is we are using. The first is in situ hybridization. Here in situ hybridization, here in this the gene analysis, where the gene is located in their natural position, that also we can analyze in this method. So next is the immunohistochemistry. In this the proteins locations we are capturing by these methods. Next, third is isolated organ perforations. It means that the separate isolated organs we are separating and doing experiment or doing research. This is one of the advantages. These are the advantages or applications of the this in situ methods. Next one is the benefits or advantages. Benefit is more physiological relevant than the in vitro, because the as it is the physiological conditions, in that only we are doing the experiments. That's why it is one of the very good advantage, more physiological relevant than the in vitro. And limitation, what are the limitations of this in situ method here? Technically demanding. That is, the technical demanding is nothing but to do this technique in situ, the expert technician requires. The everyone, all doctors or researchers can't do this experiment. The special technician requires. This is one of the limitation. Next one is the partial system views. Only the partial system view, because the only one time, this is the example rat, in that only we are separating the intestine to check the whether the in the antidiarrheal property or it is there or not. That only in the specific organ only we are checking. This is the partial system view only we can check here. Okay, that's why these are the limitations are there about this in situ method. The next one is the in vivo. Comparing to all other threes, this is one of the best preclinical methods, because the same human studies, directly we are giving the human study, the as it is animals we are using for the trial here. The in vivo is nothing but inside the living things or inside the animals, living organisms. This is the in vivo, inside the living organisms. The conducted on whole living organisms to study assess systematic drug actions. It means that we are conducting a study here on the whole living organisms to assess a systemic drug action, whether drug is acting or not, that we are analyzing on the whole living organism as it is in the living organism. This is the principle in the in vivo method. The next one, example is here the amlaki. You can take Amblica officinalis amlaki here. How we are using? Okay, you can take here the rat. In the animal studies, we have to separate take the rat. First of all, what we have to do, if you want to check the hepatotoxicity, the liver, whether the drug is toxic to the liver, the hepatotoxicity is there or not, whether the drug is affecting against the hepatotoxicity, that we are checking. Here the example is, take one rat. In that rat, we have to give this amlaki or Amblica officinalis extract, the fruit extracts that we are giving, and after that we have to give the CCLH. It means that carbon tetrachloride. Carbon tetrachloride is one of the toxic drug. That is the liver toxicity. It will produce the liver toxicity. If we are giving the carbon tetrachloride as well as the amlaki, whether this drug is protect the liver or not, that we are analyzing. Like this we are doing the experiment. Okay, this is the one of the example in the in vivo toxicity. If in their liver function test suppose normal or liver looks normal in their size shapes, it means the drug is hepatotoxic. If it is not safe, then it is considered that CCl4 carbon tetrachloride is the toxic, but it is not preventing the liver. Like that, we can analyze here using this method. This is about the in vivo method example. Next one is the applications. Application is the pharmacokinetic study. By using this method, we can analyze the pharmacokinetic study, that is the ADME, how the drug is absorbed, how the drug is distributed in our body, how the drug is metabolized in our body, as well as how the drug is excreted. All these pharmacokinetic study we can analyze using this in vivo methods. Along with the next toxicology also, whether drug is toxicity it is there or not, that also we can analyzing by using this in vivo methods. Next is the screening whether the drug is safe or not, that also we are analyzing. Next one is the pharmacodynamic evolution. Pharmacodynamics means the drug is effective or not, that also we are analyzing using this in vivo methods. The next is advantages. Why we are using this in vivo method in the preclinical study. Advantage is provide complete biological response. Okay, the whole animal we are using here, the whether complete biological response we are analyzing in this study. That's why it is one of the benefit of using this in vivo method. The next is the lastly limitations. What are the limitations of in vivo methods? In vivo method, it is a costly because we have to collect the rats and we have to go to the animal house. That's why it is costly. Ethical concerns, because for that we have to take the permission for the animal ethical committee. That's why also it is ethical concerns are there. And species difference. Lastly, species difference, because we cannot tell that the specific animals in that this drug is safe or effective. The same it will act safe in the human beings or effective as it is, we can't tell. That's why this is one of the limitations, is the specific species difference also we can analyze. This is the limitation, it is there in the preclinical study in the in vivo methods. These are the 4 methods we are using in the preclinical trial before giving to the human beings. Hope you understood the today's topic. If you understood, please share to your friends, like, share. Thank you.